enhancing the intracanal antibacterial effect of sodium hypochlorite with etidronic acid or citric acid. This ex vivo study was published in JOE 2022 by Campello, Sequeira, and Rocas and their group from Rio de Janeiro. The aim of this study was to compare the effect of 3 different irrigation regimens on the bacterial reduction. These regimens were as follows. The first one, 2.5 sodium hypochlorite. The second regimen will be using 2.5% sodium hypochlorite and alternating with 10% citric acid. The third regimen will be 2.5 and etidronic acid 10% in mixture. The latter will be mixed freshly chairside 1 hour before it is used. The rationale behind conducting such study were as follows. First, citric acid has been proposed to be used in alternating with sodium hypochlorite because it is ability to remove the smear layer. Second, in vitro study as well as randomized clinical trial showed there is an ability for the etidronic acid when it is mixed with sodium hypochlorite to reduce the bacterial number. However, the bacterial reduction has not been quantified before. That's why this study wants to investigate the bacterial reduction in numbers. 66 mandibular molars were included in this study. Teeth were accessed, and after that they were filled with tribecase soy broth, and they were placed then into microcentrifuge tube that contains the same broth. Then teeth were subjected to 2 cycles in order for the broth to penetrate into the irregularities, so it will enhance the bacterial colonization in those areas. After that, E. fecalis were inoculated inside the teeth with the incubation period of 30 days. After that, the first sample was taken. Then, teeth were prepared using reciproc blue R25, and after each 3 picking motions, irrigant was used based on each regimen as we have mentioned in the beginning of this study. Then the second sample was collected. Furthermore, activation of irrigant using XP endoshaper was done for each group. For the sodium hypochlorite group and the sodium hypochlorite group that alternated with citric acid, 1 ml of sodium hypochlorite for 1 minute was activated. However, for the group of sodium hypochlorite that mixed with etidronic acid, the same mixture was used 1 ml and it was activated 1 minute using also XP endo finisher. Then the third sample was collected. So the volume of irrigant in each group was 13 ml from the first sample to the third sample. For the results, the mixture of sodium hypochlorite with etidronic acid showed bacterial reduction, which was significant by 54% and 63% in compared to the sodium hypochlorite group and sodium hypochlorite group with citric acid after the second sample. After the third sample, the mixture of sodium hypochlorite with etidronic acid and the sodium hypochlorite group that alternated with citric acid were significantly lower in bacterial number, respectively, compared to the sodium hypochlorite group alone. However, after the third sample, there was no significant difference between the mixture of sodium hypochlorite with etidronic acid and the sodium hypochlorite with citric acid. In conclusion, the finding of this study stated the mixture of sodium hypochlorite with etidronic acid or the alternating between sodium hypochlorite and citric acid were both more effective in terms of bacterial reduction comparing to sodium hypochlorite alone. However, we need to keep in mind the limitation of this study as they mentioned they used E. fecalis here. However, if mixed biofilm used, it would be better simulate the clinical conditions.